All right. Good morning, everyone. Thanks for the opportunity to speak today. Look, today I'll be talking to you about a journey about the power of OMICS, where it's been applied to the field of extracellular vesicles, but importantly, some of the challenges and probably some of the questions that it doesn't actually address. So the field has basically had an exponential increase in the use, development and application of a variety of OMIC technologies over the last few years. And you can see this not only probably more than 3,000 sort of studies in EV track, 143 in the last year, sort of using with relevance to OMICS, okay? And also in the GEV and GEX-BIO sort of publications, also an exponential increase in the type of articles that are actually getting reported in these journals. And we know that extracellular vesicles are really complex. Complex in terms of not only their diverse luminal cargo, but also their surface composition as well. But in addition, extracellular vesicles also comprise a lot of other things than just surface and luminal components. A variety of sort of organelle fragments, DNA, and a variety of other sort of cytokines, chemokines, and post-translational modifications of those species. So what does OMICS actually help with? All right, so there's a lot of type of questions here that is geared towards potentially some of you in the audience that it may ask, you may ask what sort of proteins are localized maybe on the surface of extracellular vesicles versus sort of their luminal or even corona content. Can we understand the core elements of an extracellular vesicle, whether it's a lipid, whether it's a nucleic acid or a protein? What about markers of diagnosis? Or for that matter, what are the type of proteins or RNAs expressed in or on a vesicle? How may these actually be differentially expressed? And what about understanding biological functions or features of a vesicle? What about where they actually go? What about how their composition actually may alter over time? And can this gain clues about how they interact or target or potentially link mechanisms of how these cargo actually get in the vesicle in the first place? So, as I said, there's a variety of OMIC technologies which are behind all of this sort of field. And they sort of center around, I would say, these three main categories. And I'm only focusing for this matter around protein, lipid, and nucleic acid. So protein basically is centered around the use of mass spectrometry. There's a lot of other OMIC technologies or proteomic technologies, such as aptamer-based or even Western blotting for that matter, which is still a proteomic technology. But for the purpose of this talk, really it's a focus around mass spectrometry. It allows you to gain an insight into what are the proteins that you can identify or quantitate. What type of modifications can you look at? What about post-translational modifications such as phosphorylation or glycoforms? It allows you to look at thousands of proteins, many, many thousands. So the complexity of mass spectrometry is a hurdle in itself. You can look at global, which means as many things as possible, or you can target this and really look at a subset. The throughput, however, is a massive challenge, okay? Because you can't actually enrich a specific element, say for instance a protein, you have to go through an enrichment strategy. You can't just expand the proteome, you have to actually enrich for it, okay, and I'll explain what that means. You can also use a variety of transcript and sequencing technologies, whether this is global, such as deep sequencing for small and large RNAs. There's also RT-PCR, which is a very much more targeted sort of strategy. Again, thousands and thousands of RNA species can be profiled. You can enrich, you can expand the RNAs through a variety of sort of strategies. And the throughput of this is actually one of the key powers over the last probably 10 years of the field. There's also key advances with lipids, given that their composition is so important in the form and potential function of an extracellular vesicle. The diversity of lipid species and metabolites for that matter has really expanded over the last few years. And again, the throughput of these type of strategies is exceptional. So where has OMICS actually been applied to understand what a vesicle is and how complex it actually is? And so this is basically just a grab bag of, I guess, I would say some of the key foundational studies over the last few years that have basically addressed key elements of a vesicle, what it is and what it does. We can look at the composition or the core of a proteome, and this is a variety of key studies that have come out over the last few years. We can describe new types of extracellular vesicles or particles for that matter. We can look at the type of species in or on a vesicle that may constitute its signaling. We can look at where these extracellular vesicles are from to get an insight into phenotype or origin. We can look at their circulating composition. We can look at different subtypes, the corona, the surface, and even, for that matter, the disease subtyping. Okay, so there's a lot of power of the way in which omics has shaped the field and continues to shape the field. However, there's challenges in extracellular vesicle omics, and this is not just applied to proteomics, but actually a lot of different omics together. So one of the key challenges that we've sort of faced over the last few years is basically understanding what a specific feature is of a vesicle, all right, versus a non-extracellular vesicle. So can we start looking at what is actually constitute a specific universal feature of a vesicle versus a non-universal feature of a vesicle, or a non-EV? Some of the challenges really focus around abundance, purity, and complexity. So if you have a complex sample, you're going to get a complex message, all right? So therefore, strategies to enrich or purify and understand this complexity really is paramount to any omics sort of workflow. So here, this is, I guess, a snapshot here of the protein abundance in blood. So what we're really interested down here is this tissue leakage sort of factors or secreted signaling components. What we don't really want so much is these abundant sort of parent plasma proteins, okay? We also want a strategy that allows us to gain analytical depth and is reproducible. So when I say analytical depth, it's not so much how many proteins we're seeing, but the diversity of what we're actually seeing. All right, so if you put a blood sample in or a complex EV sample in, most of the time you're going to get abundant information out. However, if you stratify and specifically purify to understand that complexity, then you can start to understand low abundant species. So your analytical depth goes from a few hundred proteins to many, many thousand, okay? And you really want to make sure that this is consistent, not only upstream, but also the analytical downstream. Some of the hurdles of enrichment, basically the genomic sort of RNA fields, is that you have this power to amplify. And you can couple this with barcoding, so therefore you can deal with many, many different types of samples, enrich those and amplify those samples, those RNA species, very, very rapidly, and it overcomes a major, major obstacle which proteomics actually you can't do. You can't actually amplify a proteome. You can enrich for a specific subset, but you can't amplify it. So therefore, there's other approaches such as targeted or MRM-based strategies which allow you to gain subset information of that proteome, potentially without having to go through so much arduous enrichment strategies. What it doesn't actually apply to is topology. So we can't actually understand where a certain protein is localized in or on a vesicle. You have to enrich for a specific subset. And what I mean by that is that you have to look at what is on the surface, decorated surface, using, say, biotin or other sort of strategies where you can barcode this. You may have to look at a phospho-enrichment sort of strategy to understand this type of signaling components in or on these vesicles. Proteomics, it is quite complex in terms of its sample preparation complexity. Many, many different steps, many hurdles, potential many errors or sort of factors that can be introduced. So this does impact throughput. And while other sort of lipidomic and genomic sort of pipelines really is almost very kit-based, very focused sort of throughput strategies. And sample handling, storage, and degradation is a huge problem in proteomics because samples degrade, proteins degrade, and especially post-translational modifications are really, really fickle. So if you're not preserving this and you're not monitoring this, you basically will only end up with a very, very small amount of information. And obviously, reproducibility and analytical throughput is paramount for all of these challenges with EV-omics. So what about some of the omic databases that are out there, okay? And there's a lot of them, all right? And they're very powerful if you use them in the right way. So I think this has been cut off a little bit here, but there's a variety of databases such as Extracellular Vesicle and even EVpedia. A lot of diverse information with proteins, lipids, metabolites, and recently DNA species from many, many different species, okay? Recently, CF-omics, which is basically a new cell-free sort of database that has come out, focused around liquid biopsy and the power of sort of diagnostic, understanding the type of targets that you may want to select for before you even start an experiment. And recently, sort of a DNA-centric database, which is EV-add, and some of this has just been cut off just due to formatting. So these EV resources are, as I said, most of them are just a catalog, all right? You have to really look into how these studies that you really want to look for, how have they actually purified or understood their vesicles in the first place? They're from diverse species, okay? Which again, could be a potential caution to use. However, with the increase in rigor and standardization of the field, there's a lot of sort of validated protocols that are now getting built into these sort of databases, okay? And basically now generating consistent, reproducible data associated with these studies. And therefore, there's a pressing need, clearly, for homogeneous, purified, EV-centric data sets really to be used. For our own experience, basically, we use these databases to a point. But I would sort of maybe stress to use these type of things, which is basically an EV core composition. This is from cells. This is from the Kaluri group a few years ago, where they looked at many, many different cell types, purified extracellular vesicles, small extracellular vesicles from biofluids, different samples, and they basically came up with a core set. I think it was 22 sort of markers, Centinum-1 is one of them. And that's basically is what our go-to sort of composition is from cells, that we actually have a vesicle, that we have these markers, and it's consistent. And recently, from our own group, this is not a plug, but this is something that you may want to use, which is basically an app called EV-Map. And basically this means is that now we can start understanding conserved universal or hallmark features of vesicles from blood. And we can do this from either a protein-centric or a lipid-centric view. So now we can start understanding what are the core foundational features or key features of a vesicle for the first time, okay? So what about rigor and reproducibility? It's central for all of this work, all right? And there's probably three main key resources that I would steer you to. One is EV-Track. Many, many different studies, but you can start focusing around key omic elements of those studies, or key species, or more importantly, the way in which these vesicles are actually isolated and purified, as well as ranked. So really, really critical resource. Another is the MySev guidelines in 2018. And so here in table, I think number 3, there's a protein content-based characterization of extracellular vesicles. And this is really, really important if you want to start looking at inclusion markers or exclusion markers of your extracellular vesicle from a variety of sort of markers and even cell-derived sort of samples. But recently, with MySev 2023, there's sort of reporting guidelines which are really, really important to sort of adhere to. Mass spectrometry-based proteomics, minimal reporting sort of standards is really, really key. So if you're going to report a proteomic experiment, what are the key aspects that you need to actually adhere to in terms of uploading data and metadata to data repositories, as well as nucleic acid characterization, as well as lipid and metabolite sort of characterization as well. So I was asked to talk also about the translational focus. So how is omics being used in development, not only for diagnostic, but also in therapeutic sort of avenues as well? There's a recent review we've just had out, which I'll focus around this in 2 arms. One is a diagnostic arm, and the other is a therapeutic arm. So from a diagnostic arm, omics is incredibly powerful in not only understanding your vesicle, defining it, but also monitoring composition. And this is really, really important if you want to start looking at pre-onset disease or progression of this disease, okay? So here, a variety of proteomic, even SumoScan sort of studies is really pushing sort of boundaries of field, not only just in extracellular vesicle field, but clearly in many, many different other fields to do with diagnostic and monitoring. The power of machine learning, AI, and informatics is a huge element for this to control for batch or variables such as age or other sort of gender influences which do need to be monitored. The other arm for all of this is therapeutic monitoring. Okay, so how is omics actually being used in all of these pipelines to develop and monitor an extracellular vesicle therapeutic? Clearly quite key in terms of understanding mode of action. Really, really important in understanding composition. So is your therapeutic entity or entities actually within your vesicle? Can you enhance it or modify it through engineering strategies? Can you monitor this aspect through your manufacture process? Is there variance to do with batch or different sites for that matter? And can you actually start to look at potency? So this could be using proteomics or omics to not only read out composition but more importantly about how these EVs are actually reprogramming target sites such as cells or organs. The last aspect here is basically looking into the future. So translational sort of focus. Where we actually are at the moment. So a lot of clinical, a lot of therapeutic interest in this field, okay? A lot of guidelines that are being established and actually in terms of reporting are actually getting well utilized. But here we still have this key hurdle to overcome, basically this peak of expectation, so to speak, all right? So you have all of these different features of an extracellular vesicle which are itself a highlight. And you'll see here, OMICS is one of the key technologies that can actually help us understand and progress this field a little bit further. There's a lot of other bioengineering aspects to this field as well that will help in the years to come how this is actually going to be implemented. So what are the gaps in knowledge? All right, so there's still a lot, right? So if you start to look at multi-omics, and this is a term that's thrown around a little bit in terms of how do you integrate different omic data sets together, the protein or the lipid, or the protein and the metabolite. Recently, there's a cross-linking sort of study looking at protein complexes in extracellular vesicles. They're really, really important functionally. These proteins don't exist in isolation. So if you're interested, please have a look at these sort of studies. There's also a huge aspect of a lot of studies actually being centered around cells and cell-derived EVs, but there's a huge aspect of translatability when we really want to try to understand this from a systems or human level. Spatial organization. What are the entity we're looking at? Where is it localized? When is it localized? And to what level? This is really, really, really key. So a lot of key aspects in the proteomic field is really focusing now around spatial proteomics. How does the change in proteome actually is influenced in terms of subcellular organization? One of the key aspects that is still, I would say, almost missing to an extent in the field is understanding a core extracellular vesicle feature based around nucleic acids. And can we actually start understanding different post-translational modifications? So this is really, really important in not just for the form and function of a vesicle, but in terms of the biogenesis of these vesicles as well. This is really, really important in terms of endosomal or even plasma membrane-derived sort of genesis. So my last slide basically here is basically looking to the future, and I've focused this around two elements. One is interactomics, so multi-omics to an extent. So how is different types of omics, so how is a protein impacting, for that matter, a lipid? So for instance, an enzyme, how is it actually impacting a lipid or a variety of lipids? How is a kinase or an enzyme impacting the phosphorylation landscape? And they're two sort of key examples. So what about next generation technologies? So in terms of proteomics, which is a lot of our field, there's been a few key advances in the last 2 to 3 years of instruments allow you to go into incredible depth that we can't actually, you're acquiring huge amounts of data without having to enrich and specifically purify samples before. So if you start looking at single cell sort of proteomics and single cell fields, this is basically where we're pushing at the moment. The next is spatial organization. So how is something actually organized at a spatial setting? Aptamers has been raised a few times already in this meeting, but this is something that actually allows you to specifically target and understand, and then power with omics to really define. Spatial organization, where is something localized, when is it localized, and how is it actually impacted in terms of localization? And last, basically, this is a key study that's come out recently where you can actually start looking at single, not so much EV, but single cell transcript remodeling to do with EV treatment. And this is really, really important if you start to look at EV function at a single EV type level. So this is just a few features of OMICS. If you're interested at all, come and have a chat. Any questions, I'm happy to answer later. Thank you.